The connection between the gut and the brain is no longer a speculative idea. It is rapidly becoming one of the most promising frontiers in mental health research. At the heart of this shift is the realization that the gut does more than digest food. It actively communicates with the brain in real time using neural, hormonal, and microbial signals. Central to this communication is the emerging role of neuropods, specialized sensory cells in the gut that translate microbial activity into rapid, neuron-like signals that travel up the vagus nerve to influence brain function. As psychiatry continues to grapple with the limitations of conventional treatments for mood disorders like depression and anxiety, the gut-brain axis is offering a new framework, one in which mental health is shaped not just by neurotransmitters in the brain, but by messages that originate in the gut. The idea that microbial signaling can affect mental health has gained support from several converging lines of evidence. One of the strongest comes from animal models. Germ-free mice, those raised without any microbes, show exaggerated stress responses, lower levels of brain-derived neurotrophic factor, BDNF, and abnormal serotonin signaling. These mice also display behaviors analogous to human anxiety and depression, such as social withdrawal, anhedonia, and hypervigilance. Reintroducing a healthy microbiome can reverse many of these behaviors, suggesting that microbial presence isn't just a modifier, it's a requirement for normal emotional development and stability. But how do microbes in the gut exert such profound effects on the brain? This is where neuropods enter the picture. These neuron-like cells sit at the interface between the gut lumen and the nervous system, equipped to detect microbial metabolites such as short-chain fatty acids, SCFAs, tryptophan derivatives, and bile acids. In response, they release neurotransmitters like glutamate, ADP, or serotonin directly onto vagal afferent neurons, creating a fast, hardwired pathway for gut information to influence the brainstem and beyond. This is a radically different view of microbial signaling, not as slow-acting background chemistry, but as high-speed sensory input that actively shapes the brain's mood circuits. Depression, for instance, has long been associated with altered serotonin levels, but more recent insights suggest that gut-derived serotonin production, which accounts for roughly 90% of the body's total serotonin, plays an underappreciated role. While most of this serotonin does not cross the blood-brain barrier, it influences the availability of serotonin precursors and modulates the vagus nerve, indirectly shaping central serotonin dynamics. Furthermore, certain gut bacteria can influence the expression of enzymes like tryptophan hydroxylase, which regulates how much tryptophan is shunted towards serotonin versus kynurenine pathways, the latter being associated with neuroinflammation and depressive symptoms. Neuropod-mediated communication is also thought to influence stress regulation, particularly through the hypothalamic-pituitary-adrenal (HPA) axis. Chronic stress alters gut barrier integrity, shifts microbial populations, and reduces microbial diversity, all of which can distort the signals neuropods transmit to the brain. In turn, maladaptive brain responses, such as elevated cortisol or heightened amygdala activation, can reinforce gut dysfunction, creating a vicious feedback loop that sustains anxiety or depressive symptoms. The vagus nerve offers a potential break in this loop. Stimulating it, either electrically or indirectly through behavioral practices, has been shown to reduce inflammation, increase parasympathetic tone, and improve emotional regulation, all of which are beneficial in mood disorders. What makes this field so compelling is its therapeutic potential. Unlike many psychiatric medications, which target neurotransmitters directly in the brain and often come with side effects or limited efficacy, interventions aimed at the gut may offer holistic and safer alternatives. Early clinical studies show that probiotic supplementation, especially with strains like lactobacillus and bifidobacterium, can modestly reduce symptoms of depression and anxiety. These effects are thought to arise not just from microbiome changes, but from downstream shifts in vagal tone, immune activity, and neuropod-driven brain signaling. Similarly, dietary interventions that enhance microbial diversity, such as high-fiber, polyphenol-rich diets, have been shown to improve mood and stress resilience, particularly when maintained over time. Even the inflammatory model of depression, once seen as separate from gut health, is now tightly linked to microbial signaling. Dysbiosis can lead to increased gut permeability, leaky gut, allowing lipopolysaccharides and other pro-inflammatory molecules to enter the bloodstream and activate immune cells in the brain. This neuroinflammation is strongly associated with anhedonia, fatigue, and treatment-resistant depression. By contrast, a well-balanced microbiome can reinforce gut barrier function, suppress excessive immune activation, and help maintain a low-inflammatory internal environment, all of which are neuroprotective. The role of neuropods in this framework cannot be overstated. These cells act as gateways of perception, converting gut chemical states into language the brain understands. Fast, synaptic communication. This suggests that how we feel, emotionally, is not only a reflection of thoughts or circumstances, but also of how well our gut is perceiving and reporting on the world inside us. When this system is functioning optimally, it creates a state of emotional flexibility and resilience. When it's disrupted, the brain may respond with chronic anxiety, despair, or irritability, even in the absence of external threats. In conclusion, the emerging science of gut neuron communication is not merely adding detail to existing psychiatric models, it's rewriting the foundations. It challenges the brain-centric view of mood disorders and offers a richer, more systemic understanding of mental health. As neuropod research advances, it may allow us to develop new treatments that don't just suppress symptoms, but restore the integrity of the brain-body dialogue itself. For patients and clinicians alike, this marks the beginning of a more integrative and hopeful chapter in psychiatry, one that listens to the signals not just of the mind, but of the gut that speaks to it.